Hello everyone, I am Emila Panda. I am Associate Professor in the discipline of Materials Engineering at IIT Gandhinagar. I am here for past 10 years. Before joining here, I did my PhD and postdoc in Max Planck Institutes in Stuttgart and Halle respectively. Here at IIT Gandhinagar, my group is primarily responsible in fabricating thin film coating for various kinds of applications. One of the major application is optoelectronic materials, including transparent conductors and thin film solar cell. As a secondary objective, our group also works on oxidation phenomena and tries to understand the thermodynamics and kinetics of various oxide phases developed on magnetic materials as well as other kinds of materials. In this project, I am going to talk about highly reactive, transparent and environment-friendly antimicrobial surface coating technology. The team members who actively participated in this innovation or project are Mr. Miti Reddy Rabiteja, PhD student in Materials Engineering, Dr. Nishaben Patel, past doctoral student at Biological Engineering discipline, Dr. Virupakshi Sopina, Ramalinga Swami Fellow, and Dr. Abhay Raj Singh Gautam, Assistant Professor in Materials Engineering discipline at IIT Gandhinagar. So, coming to the motivation of this work, as we know, the microbe-induced diseases have led to a significant rise in the infection rate, causing widespread morbidity, mortality, as well as disruption to regular activities. So, many of these microbes, for example, the coronavirus, could be highly contagious and remain viable on solid surface for several days to weeks. And the primary means of contamination and the spread of microbes has been always a human who comes in contact with the solid surfaces, and because on these solid surfaces the microbes can be viable for several days. So this requires synthesis of surface coating that can effectively inactivate the microbes, and the surface coating need to be prepared easily, should be economical and non-cytotoxic, should have strong adhesion with the substrate, so that means it should not come out from the substrate easily, should be durable, and more or less it should be transparent, because if it is transparent then it would not compromise the aesthetic aspect or design aspect of particular material, and then it would be easily adopted by the customer or end user. Coming to the range of products which are currently available, so these are basically some of the products you can see. Mostly or in general, these products these products consist of toxic opaque layers, mainly consisting of copper and silver based materials. So if you look at it, so here is a mask on top of which this coating has been applied, and as you can see, the color of the coating has been changed. Top of that, this consists of silver nanocluster, which is toxic. So what we did here at IIT Gandhinagar, we have basically created a surface coating which consists of amorphous non-stoichiometric version of titanium dioxide. So this is how it looks like. So you can see this is glass, and you can basically have this coating, surface coating on any kinds of surfaces at room temperature. So this is a coated glass. How does it look like when we have done a 10-minute deposition, and here another where we have done a 15-minute deposition. So basically the properties do not change. And if I can show you, so this is how basically it looks like. So this is really transparent. This coating has been developed on the glass substrate, and this is also an aluminum substrate on top of which the coating has been developed, and you can see that it has not changed. So similarly, you can see on silicon and on Teflon, the nature of the base material remains unchanged. This is how the optical transmittance looks like, and you can see that the material has very good transmittance. We have also done the delamination test, that means using the tape test, So we found that the material is the coating is very stable, it doesn't come out of it. We have also used strong detergent test using the ISO 4794 standard, and we did not actually see much change in the color. So it says that the coating is really stable to strong detergent. Coming to the results of the anti-microbial, so this is how the antibacterial activity looks like. So here you can see that the number of colonies are really high on plain glass substrate. Here we have tested it against E. coli, but if you look at on the coated substrate, you can see that the number of colonies have drastically reduced. So it shows a very high antibacterial activity of this surface coating. Similarly, if we look at an antiviral activity, so we tested it against baculovirus, and the green fluorescence proteins basically says indirectly the number of viral strains present on the sample surface. You can see a large number of green fluorescence proteins on a glass substrate, and you almost see nothing here on the surface coated glass, so which says a very high antiviral activity again. We also washed the surface several times, and we hardly saw any change here. So it shows that the coating is very stable. We then tested the cytotoxicity of this coating against several cell lines, HeLa, SF9 and CAD cells, and like you can see the cell viability, so these are extremely non-cytotoxic. So in general, we have now tested against a range of microbes like envelope as well as non-envelope type of viral strains like Newcastle disease virus, baculovirus. We have also tested against gram-positive and gram-negative bacterial strain like SRS and E. coli, and pathogenic strains present in seawater, and this was the patent we file now. So coming to the current technology readiness label, so this innovation is a TRL5, and it has a significant market. That's because the surface coating can be developed on any kinds of surfaces, starting from plastic to tile to glass to metal or alloys, and therefore it could be a significant interest to any glass, tile, bioimplant, ceramic manufacturer, etc. It has a huge societal effect. That's because this developed surface coating would be able to control and mitigate a range of microbes, and this could have a significant positive effect on the public health. Coming to how do we increase the TRL label, so this is just a schematic about a continuous pilot plant or a demonstration plant which can be set up for demonstrating the technology at a large area and with a high throughput. In this process, we would be able to scale up the process, and we would be then able to do a complete validation for its economic feasibility. So the technology has reached TRL5, and like I have already mentioned, this coating can be developed on any kinds of surfaces, be it wood to plastic to glass, ceramic, alloy, metals, and at room temperature without changing its aesthetic. That means it is transparent, and like I have already mentioned in the previous slide, the coating is non-cytotoxic. That means this is not harmful at all to human or animals, to any cell lines. However, this is very toxic to the viral as well as different bacterial strains, and therefore if we referring to the current pandemic which we all are facing now, so if we have such kind of a coating in place, then this would be able to inactivate the viral as well as some of the bacterial strains in hospitals, and could have significant positive effect on the public health.